Okay, so we have here the formation of your ABH substances or ABH antigen in your RBC membrane. So again, you're needing here the mga genes, your H gene, and we have also your ABO na genes. Except your O na gene, wala ka, again, remember that your O gene is the amorph na gene, and therefore that one do not code here for the production of particular antigen. Okay, so the first one, so we have here your H gene. So again, ang mga gene natin, nagko-code here, I try to code for the production of our enzymes. So ito mga enzyme na pinoproduce nila. Okay, your H gene, okay, try to code here for the production of your alpha 2L fucosyl transferase. And this is the enzyme which try to transfer the sugar L-fucose, which is the immunodominant sugar here in your H. Okay, immunodominant sugar here for your H na antigen. Okay, so they try to transfer here the L-fucose to your type 2 precursor. Ito yung type 2 precursor natin. Okay, your type 2 precursor, the paragloboside, so this is the RBC membrane. So, pag sinabing type 2 precursor, the globoside, ito yung mga ano natin, parang made up of the sugar moiety. So, glucose, yung pinakamalapit sa RBC membrane, followed here by the D-galactose, may naka-attach na N-acetylglucosamine. Then, we have your terminal D-galactose na sugar. And the entire structure, you call this one as your type 2 precursor or the paragloboside. So it's being linked here by your beta 1,4 linkage. The beta 1,4 linkage try to link here the terminal D-galactose to your N-acetylglucosamine. It's beta 1 because your galactose, both of them would have here 6 mga hexos ito, diba? So these are 6 carbon sugar. So, your D-galactose, the carbon 1 nito, naka-attach kay carbon 4 ni N-acetylglucosamine. Kaya tinawag siyang beta 1,4 linkage. And this is again your type 2 precursor. So, sa H gene natin, itry to code here for the production of the enzyme alpha-2L-fucosyl transferase na mag-a-add siya ng L-fucose dito sa ating terminal D-galactose sa pinaka-end part para makapag-produce ka ng H antigen. On the other hand, for the A na gene, try to code here for your enzyme N-acetylgalactosaminil transferase. We try to add N-acetylgalactosamine dito para makapag-produce ka ng A antigen. For the type B naman, they have ang kanilang B gene. Try to code here for the production of your enzyme na galactosyl transferase. We try to transfer your D-galactose dito sa pinakahuling D-galactose, i-add niya para makapag-produce ka ng B na antigen. Okay, your H gene, para makapag-produce ka ng H antigen, dapat ang ma-inherit natin na H gene would be homozygous for the H, dalawang malaki, or heterozygous for the H, isang malaki, isang maliit. Again, para makapag-produce ka ng H antigen. Pag ito ang na-inherit mo, homozygous, okay, dalawang maliit, hindi ka makapag-produce ng H antigen. Okay, clear? Pag hindi ka makapag-produce ng H antigen, hindi ka rin makapag-produce ng mga ABO or AB antigens. Mean to say, kailangan mo munang H antigen para makapag-produce ka ng A antigen saka B antigen natin. So kung dito pa lang, ito na kaagad ang iyong H gene, okay, homozygous for the small, dalawang H na small, and therefore wala kang H antigen na maproduce. Pag wala ka ma-produce na H antigen, so wala ka din dito, hindi ka rin makapag-express ng A or even the B antigen in your RBC membrane. Okay, clear? Okay, so if you are type A, dapat you need to inherit this one. Okay, so either of this one. So you have your H antigen for that. And then you need to have also here the A na gene. Okay, that would add your N-acetylgalactosamine here. Okay, para ma-change ang ating, okay, so ano nangyari? Ulit, if you're type A, for example, if you're type A, number 1, kailangan mo ng H gene. Okay, your H gene should be homozygous for your H, na dalawang H na malaki or heterozygous for that H na malaki saka maliit. So, if you have this gene, okay, and eventually, you're going to have here your fucosyl-transferase na mag-a-add ng L-fucos. So, mag-a-add dito ng L-fucos dito sa taas. Pag nag-add na siya ng L-fucos, the entire structure that become your H antigen. Okay, so therefore, may H antigen ka na. The next one, if you're type A, kailangan mo you have your A na gene. And the A gene here try to code for your N-acetyl galactosaminyltransferase, which add niya ito ulit. Okay, dito sa ating nakatouch na L-fucos, i-modify na ito and eventually converting your H antigen to your A antigen. And therefore, pag A ka, anong antigen natin? H una, followed by the A antigen. Okay, for the type B naman, so ulit, kailangan mo ng H gene, which is dapat ito saka ito, para makaproduce ka ng H na antigen. Then, kailangan mo ng B na gene, which try to code here for your production of your D-galactosyltransferase para mag-add ng D-galactose dito ulit para i-modify niya itong H antigen mo para magiging B antigen. And therefore, for the type B, you have H and a B na antigen. How about for type O na individual? Okay, pag true type O individual, meron pa rin siya, kailangan mo pa rin ito ng H. Meron siyang H antigen. Okay, so first, for the true type O, so kailangan mo ng H gene. So this one, homozygous for this one, or heterozygous for that one. For the production of your, ganyan, mag-add L-fucos, this become here your H antigen. So therefore, true type O still have here the H pa din na antigen sa kanyang RBC membrane. Since ang ating, wala tayong O na gene, di ba? May O tayo na gene. However, your O na gene is amorph. Since amorph siya, mean to say, it will not produce antigen. Wala naman tayong O antigen, di ba? Kasi amorph ang O gene natin. And therefore, hanggang H antigen lang ang ating true type O individual. Okay? Okay, example for that we have here. For example, if the patient try to inherit here the H gene, having this homozygous for the H, and meron siyang A sa B na gene. Okay, mangyari niyan. So, if you are asked here, what are the ABH substances found in the RBC membrane in the patients? Number 1, may H gene ba may H antigen ba siya? Yes. Kasi this one is your homozygous for that. So, we have your H gene. So, A antigen, we have also here the B antigen. Pero pag ito, again, pag maliit, kahit may mga ABO gene ka pa din, hindi pa rin, again, pag wala kang H gene in the form of your heterozygous for that, hindi ka mag-express or pag hindi ka maproduce ng H antigen, you will not also produce here your ABO na mga antigen. So pag ito ang kanyang genotype for the H, so mean to say wala kang H na antigen na. So, wala ka ng H antigen, hindi ka mapaproduce ng ating ABO antigen regardless kung meron ka nito. Kaya wala ka talagang ABH substance in the RBC membrane. Another one ito, HH. So, dito pa lang, wala kang H na gene or H antigen na maproproduce. So, therefore, wala ka ding antigen, ABO antigen. Pero pag ito, may H antigen ka, since ang O gene natin is amorph, wala kang ABO na antigen, therefore H antigen lang present or maproproduce natin or present in the RBC membrane of the patient. Okay, another one, we have here your ABH substance found in our secretions. The ABH substance found in our secretions, the production of that is under the influence of your secretory gene, which is also encoded in chromosome number 19, just like our H gene. Okay, so the secretory gene try to code here for the production of your secretor, also for the production of your H na antigen. Pag nasa RBC membrane, ang H antigen natin is under the influence of your production of your H gene. H antigen is under the influence of your H gene para makaproduce tayo ng H antigen in our RBC membrane. Pero, the production of your H antigen in your secretion is under the influence of your secretory gene. So, para makapag-produce ka ng H na gene, or H antigen, in your secretion, kailangan mo ng secretory gene. And you should be heterozygous for this one or homozygous for that. And not yung dalawang maliit. Pag dalawang maliit, hindi ka makapag-produce ulit ng H antigen. Yung secretory gene also code for the same sugar ng ito din ang sugar ni secretory gene natin. Alpha-2 L-fucosyltransferase, mag-a-add siya ng L-fucos ulit to your type 1 precursor. Type 1 precursor, ganito pa din siya. Glucose, D-galactose, N-acetyl, glucosamine, terminal D-galactose. Only that, instead of your beta-1,4, beta-1,3 linkage, ang ating type 1 precursor. Okay, so type 1 precursors for the production of your H antigen in your secretions. Pero nag-influence na para mag-produce tayo ng H antigen in our secretion is your secretory gene. Then, para makapag-produce tayo ng mga ABO, ng ABO na substances in our secretions, kailangan mo muna makapag-produce ng H gene. So, kailangan mo ng secretory gene. Okay, so ulit, if you try to inherit this one, ganito, homozygous for this one, and you have your A. So, again, your secretory gene, we're going to code here for the production for H antigen, and we have your A pa din siya, A antigen. Therefore, what you have your H saka A antigen in your secretions. Ito naman, so since you have secretory naman, so that's H gene, and A and a B gene, and we have your H antigen, because of this one, and your A correspond to your A antigen, B for your B antigen. Pag ito ang na-inherit natin na secretor, ito homozygous for this one, maliit. Dito pa lang, wala ka ng H antigen. So, kahit na meron kang gene dito for the A, wala ka pa rin mapoproduce na antigen sa ating secretion kasi dito pa lang maliit ng ating secretor. Okay, then we have here your correlation for that. Like, for example, if you have this genotype, so we have HH, we have also here your heterozygous for your secretory, we have your AB. So, again, if you are talking about the ABH substance in the RBC membrane, so ito ang kinoconsider dapat natin. So, since meron kang H, di ba? So, it could express this one as your H antigen. So, therefore, what you have is H antigen. And still, you have your AB onogen here. So, A and B. So, those are the ABA substance found in our RBC membrane. How about in the plasma? In the secretions. So, ABA substance in your secretion here is under the influence of your secretory gene. So, dito ka titingin. So, since you have this one, heterozygous for your secretory gene. So, still have your H antigen because of that. Okay, then we have your, since meron kang A, so you have your A pa din and B. So, H, A, and B pa din makikita natin sa ating secretions. On the other hand, if you have this one, okay, na genotype, so in our RBC membrane, ito at sa tayo ulit tingin, first ito sa H. This one is homozygous for the H. This one will not code for the production for H antigen. So, therefore, pag wala ka ng H antigen, wala ka ding mga ABO antigen. So, therefore, wala ka dito. Here naman sa secretion natin, ang titingnan natin is secretory gene. So, secretory gene natin is also homozygous for this one na dalawang maliit, and therefore, wala kang H antigen. Pag wala ka ng H antigen, wala na. So, kahit pa-proceed ka pa doon, kahit may ABO gene ka pa, or A or B na gene, wala pa din na puproduce. So, therefore, wala. Here naman, so we have here your, okay, homozygous, so RBC, ABH here in your RBC membrane. So we have that, since one, this is your H antigen, or H gene, or H na genotype. Okay, so this will code here for the production for H. Therefore, you have your H antigen. Pero your O, since amorph ito, wala kang ABO na gene, so H antigen lang tayo in the RBC membrane. How about in your secretion? So, titignan mo ulit ito. This one can produce the H antigen. So, may H ka. Since ito ay O ulit, so hindi ito magkocode ng production ng O antigen. Therefore, H antigen lang meron ka. On the other hand, ito naman, So, in the RBC membrane, so itong titingnan muna natin. So, this one could code here for the production for H antigen. And since may B na ka na gene, so still have here the B antigen in the RBC membrane. In the secretion, since ito ang ating secretor, dalawang maliit, it will not code for the production of H antigen. Pag wala ka ng H antigen, yung secretion, wala ka na ring mga ABO or AB na mga antigens. Therefore, wala ka na lahat on your secretions. Okay, then we have here the difference between your ABH substance in our RBC membrane versus your ABH substance found in our secretions. Okay, the ABH substance in our RBC membrane could be the form of your glycolipids, glycoproteins, or glycosphingolipids. But in the secretions, our ABH is only glycoproteins. Another one, in the production of your ABH substance in your RBC membrane, you are requiring to have the type 2 precursor. And that's why the terminal D-galactose is being linked to your N-acetylglucosamine by your beta-1,4 linkage. In secretion, so ABH production is under you need here the type 1 precursor and linked here by the beta-1,3 linkage. Okay, for the production of your H antigen in your RBC membrane, kailangan natin ng H gene. And for the production of your H antigen in your secretion, kailangan natin ng secretory gene. Both of that are encoded in a chromosome number 19. Your secretory gene and H gene are not part of your ABO blood group system, but they tend to influence the production of your ABH antigens. Okay, then we have here the relative concentration of your H antigen. So, pinakamadami is your O. Type O, madami siyang H antigen kasi, di ba? Hindi naman na-modify ang ating H antigen for type O. Hindi siya nag-change, di ba? Kasi wala ka namang, again, your O gene is an amorph. So, second here na pinakamataas ang concentration ng H antigen, A2, followed by your B, A2B, A, and finally, we have your A1B. And then we have here your lectin. Pag sinabing lectins, these are your plant extract that would eventually try to react with your carbohydrates. So, ginagamit natin itong mga anti-sera. Okay, so parang antibody ito para mag-detect ng ating antigen. So, para ma-detect ang ating A1, pag nag-add ka ng dolichos bifluoros, pag nag-positive, meaning to say ang patient natin may A1 na antigen. Your Bandereia simplicifolaria would eventually be detected your B na antigen. And your H antigen could be detected here by your Yolex europaeus. Again, mga anti-sera. Ito ay mga antibody para ma-detect ang mga antigen na ito. Okay, now we go to your weaker reactions. So, we have here the weaker mga subgroups natin. So, the subgroups could be identified here by having a weaker reaction with the usual na blood typing. Like, for example, if you're type A, pag nag-add tayo ng anti-A, anti-sera, dapat 3 plus or 4 plus ang kanyang reaction. So, pag mga 2 plus, 1 plus, or something like that, weaker reaction, then we are suspecting for the presence of our subgroups. Okay, the first subgroups here, we have your subgroups for your A, divided into your A1 and your A2. Okay, so your A1 and the A2, so if you have your anti-A, anti-B, so ito yung ating forward typing where we are detecting for antigen. So both of them will give a positive reaction here for the anti-A. Pag na-positive dito, signifying na meron silang A antigen. Anyway, they are type A naman, kaya both of them would have the A antigen. Anti-B, anti-sera, na reagent na negative sila. So, because again, they are type A. Okay, now we have here the antibody. Okay, so, okay, for the antibody, so ito ang ating antibody. Okay, you could have here your okay, we have your A1 cells. Ay, negative din ito. Wait lang. Okay, we have your A cells. Okay, the A cells here, so this would detect for the presence of your anti-A antibody. So both of them will give A negative result kasi di ba they are type A. So wala silang anti-A antibody. So pag may B cells tayo which would detect the presence of your anti-B antibody, both of them will give a positive reactions. Why? Because they are A naman, di ba? Pag type A, may anti-B antibody. So that's why positive dito sila. So ang antigen na makikita natin, for the A1, meron siyang H antigen, A antigen, plus meron siyang A1 na antigen. For A2 naman, ang antigen niya lang ay H saka A antigen. Wala siyang A1 na antigen. Okay, so we have here antibody. So, expected antibody, since they are type A, na-expect natin na meron silang anti-B antibody, anti-B antibody. But then again, ang A2 natin, since wala siyang A1 na antigen, pag wala kang A1, so magde-develop siya ng anti-A1 antibody. Ang A1 natin, wala siyang anti-A1 antibody kasi may A1 antigen siya. So, pag may A1 antigen, so wala kang anti-A1 antibody. Ito naman kasi wala siyang A1 antigen, so you're expecting to have anti-A1 antibody. Paano natin madidetect kung sino yan ang merong A1 na antigen? So, gagamit tayo ng lectin na anti-A1 lectin. So, anti-A1 lectin will detect your A1. Ano ulit ang A1, anti-A1 lectin natin for detection of your A1? That's your dolichos bifluoros. So, pag gumamit ka ng dolichos bifluoros, that's yung reagent mo na yan ay anti-A1 lectin para makadetect ka ng A1 antigen. So, ang may A1 antigen lang natin is your A1. So, siya lang magpa-positive pag nag-add ka ng dolichos bifluoros. Ang A2 natin, magne-negative siya kasi wala naman siyang A1 antigen. Again, your A1, okay, ang A1 natin, meron siyang H antigen, A antigen, saka A1 antigen. Ang A1 antigen natin, madidetect pag nag-add tayo ng dolichos bifluoros. Ang A2 natin, may H antigen siya, A antigen, pero wala siyang A1. So, therefore, pag nag-add tayo ng dolichos bifluoros, ang A2 natin, magne-negative siya. For the antibody, your A1 will have here anti-B antibody lang. But your A2, meron siyang anti-B antibody plus anti-A1 antibody. Okay, then we have here the difference between your A1 and your A2. So, the first one, we have here prevalence. So, majority of the type A na mga individual, almost 90% are type A1. The remaining percentage are your mga population are A2 sila. Another one, antigenic site. Madami antigenic site ni A1 compared to your A2. And for the H and the A na mga subgroups here, so your A1, so we have here AA, B, AB, AC, AD. Wala kang H, unconverted na H. Okay, kasi lahat meron ka A, AB, AB, AC, AD, so walang H. Dito naman, ang meron lang sa kanya AA saka AB. So, may unconverted siya na H3 saka H4. Kasi remember your H antigen, mako-convert to your A antigen. Like for example, your H1, ganito kasi H1, H2, H3, H4. Okay, ang procedure natin ulit, ang H natin, para makaproduce ng A, mako-convert sa A natin. So, ang H1 mo makonvert to your AA, H2 makonvert to your AB, H3 makonvert to your AC, H4 makonvert to your AD. So, since meron ka dito sa A1, AA, BB, AC, AD, mean to say, lahat ng mga ito ay na-convert lahat. Kaya wala kang unconverted na H. Dito naman sa A2, meron lang siyang AA, saka AB, mean to say H1, saka H na-convert. So, ang H3 saka H4, hindi na-convert. So, the remaining is H3 and H4. That's would have here your A2 na individual. Then we have here another mga weaker subgroups for your type A. Okay, so if you add here your anti-A, anti-A1B na anti-sera, they will give a weaker reactions. Example for that, we have your A3, A and AX. So they identify here by A3, more than 50% population here. Pag nag-add ka naman ito, magkakaroon siya ng mixed field reaction. Pag sinabing mixed field reaction, Pag tinignan mo ang kanyang sample ng patient upon addition of this reagent, may mga part, okay, may mga area na magkakaroon ng agglutination, may mga area na walang agglutination. That's your mixed field agglutination reaction. Majority of that, almost 50%, could be found in the A3 na subgroups. AN, on the other hand, would have here only less than 10% of the mixed field reactions. Your AX, ito ay magre-react lang dito sa antisera natin. Hindi siya dito magre-react. Okay, then we have here in their secretions, or even in their RBC membrane, okay, ang kanilang antigen makikita natin H saka A for your A3. AN and AX, H lang ang makikita natin na antigen, both in the RBC membrane and their secretions. Okay, antibody, since type A sila, we're expecting that they have an anti-B antibody. Anti-B ito, anti-B antibody. So, unexpected antibody. Since mayroon siyang H saka A, okay, so wala tayong unexpected antibody. Ito naman, H lang kasi siya. So, may tendency na mag-develop siya ng anti-A antibody as or unexpected. Ito naman, okay, so since H lang makita natin sa RBC membrane niya, sa kasama secretion, may tendency siya mag-develop ng anti-A antibody. On the other hand, we have also another subgroups, AM, AY, AL. So, pag itong ginamit mo na mga reagent, they will not give a positive reactions. Not unless you perform the adsorption and the elution na test. Pag adsorb, okay, so kung itong, pag RBC natin ito, pag nilagay mo ng antibody dito, try to attach, that's your adsorption. Pag tinanggal mo naman siya, that's your elution. So, ang mga ito, hindi sila magpa-positive reaction with just addition of your anti-A sa anti-AB. So, kailangan mo munang mag-perform ng adsorption sa elution in order for them to be reactive. AM, AY, AN. Okay, they have here their antigens found in the RBC membrane and their secretions. For the AM, H antigen sa A antigen, AY is A and H, and AL is only H. So we have their expected antibodies. Since type A sila, you're expecting they have an anti-B, antibody, anti-B, antibody, anti-B, antibody. So ito, unexpected antibody. So since doon na siya H saka A antigen, wala kang unexpected. Ito H and A naman, so wala kang unexpected. Ito ay walang A. So therefore, may tendency siya ma-develop ng anti-A antibody as the unexpected antibody. Okay, then we have also here the B subgroups. We have here the B, we have your B3, BX, BM, and we have your BL. Nasa book naman nyo ito. So, we have here your forward typing with addition of anti-A, anti-B. So, all of them will give a negative result here kasi wala naman silang B antigen, wala naman silang A antigen. Addition of your anti-B, anti-sera will give a positive reaction for them. Okay, so for the true type B here, we have here 4 plus B3BX would have here a B3 would have mixed field reaction, BX weak reaction, BM mixed field reactions, and BL would have a negative reaction. Antigen found both in their RBC membrane and their secretions. Okay, true type B here we have H and a B, B3 we have H and B, BX H lang, BM H and B, and BL we have here H. We have your expected antibodies. So, since all of them are type B, ang type B natin may anti-A antibody. So, lahat sila. But notice here, they're unexpected antibody. So, for your B, since may H and B siya, so wala kang unexpected antibody. Same true with your B3. How about your BX? Since ang antigen nila ay H lang, so you might expect here some weaker anti-B antibody. So, ito din, since may H saka B, so wala ka dito na expected antibody. Ito naman, since may H antigen lang meron sa kanya, weaker action for the B antigen, so therefore, it might expect here to have the presence of your anti-B na antibody as unexpected na antibody. Okay, now we go to your Bombay phenotype. So, Bombay phenotype here has been named here as the Bombay because this one has been discovered here. The first case was then discovered in Bombay, India. So the Bombay phenotype would resemble here our true type O. True type O, Okay, would have here the H antigen lang na present sa kanyang RBC membrane and in his secretion. But the antibody present sa kanyang secretion, sa kanyang plasma or kanyang serum, so type O siya, so lahat ng antibody, anti-A, anti-B, and anti-AB. Anti-AB. For the Bombay phenotype, anong pinagkaiba? Ang Bombay phenotype natin, wala talaga siya kahit H. Okay, so ano ito? Parang null phenotype siya. Ganon, HH. So ito ay, okay, OH. Ganon. Ito ay ganyan, OH na maliit. So therefore, wala siyang H antigen found here in the secretions and even the RBC membrane and the Sir Bombay phenotype. Since wala siyang H antigen, we are expecting here na magkakaroon siya ng anti-H antibody. Yan ang pinagkaiba niya sa true type O natin. And then for the Bombay phenotype, we have here anti-A, anti-B, anti-A1B, and anti-H antibody. Okay, so for you to differentiate if that one is a true type O and or Bombay phenotype, so mag-add ka ng lectin na Yolex europaeus. Yolex europaeus is antibody, is a lectin serving as antibody for detection of your H antigen. This is for detection of your H antigen. Pag nag-add tayo ng Yolex europaeus, pag nag-positive siya, mean to say ang sample natin ay may H antigen. So, sino sa kanila may H antigen? Okay, only the true type O ang may H antigen. So, ang magpa-positive lang sa Euler surveys natin would be your true type O but not your Bombay na phenotype. Okay, so true type O natin ay may H antigen, wala siyang anti-H antibody. Ang Bombay phenotype natin may H, wala siyang H antigen, and therefore it would have the anti-H antibody. Okay, so anti-H antibody is an IgG antibody. So it's clinically significant antibody reactive at 37 degrees Celsius. So that's your body temperature, making that one as highly dangerous. So, anong maging implication nito? If the patient is Bombay, hindi mo dapat siya sasalinan ng blood coming from true type O. Why? Because anong rule ulit natin para mag-compatible ang donor sa recipient natin? Ang recipient natin dapat ay walang antibody corresponding to the RBC antigen of your donor. So notice here, kung ang recipient mo, for example, ang salinan mo ay Bombay, ang Bombay natin ay may anti-H antibody. Okay, so ang ating type O na individual here ay may H antigen. So may antibody ka na anti-H with your recipient, may H antigen ka with your donor. So magkakaroon siya ng hemolytic reactions. That's why be careful here. Kasi pag hindi ka nag-perform nito ng anti-H lectin mo, hindi ka mag-add ng anti-H lectin for detection of your H antigen, baka mapagkamalan mo sila. Like routine lang na blood typing, anti-A, anti-B lang, wala kang anti-H na lectin. So pareho sila mag-appear na type O. Okay, so it's because they will not have here reactive sa A, anti-A na anti-sera kasi wala naman silang antigen A, or anti-B, anti-sera, wala naman siyang anti-B, o wala naman siyang B na antigen. So, kailangan mo gumamit ng, okay, especially pag mga foreigner ang ating patient. Okay, because again, hindi pwedeng salinan ang ating Bombay phenotype ng true type